you very much for the invitation. It's nice to be here. So today I'm going to try to tell you our latest developments on the new techniques to assess these patients. And this is a nice figure that we created a bit to showcase our problem. It's like, you know, you're facing this dilemma of someone who's completely silent, in the sense that gives you no evidence of what's happening inside, and then you try to create all sorts of techniques to assess, in fact, if this person is, you know, conscious, connected to the external world or not. So it's a nice figure we propose to a cover of an article that's coming in a couple of weeks. Let me first, you know, try to explain a bit the motivation behind the line of research that we are following. So our first big important question is, what drives the dynamics of our subjective experience? And for that, there are two types of questions, two lines of research that most of the people do. One line of research is the study of states of consciousness. The other, it's about the contents of consciousness. So when we talk about states of consciousness, it's a global description of an individual or an animal, or, you know, perhaps one day a synthetic system, in which we say whether this person is awake and aware, is connected, he can respond to our questions. I don't care about what's going on his specific mind. So that's what would be one global state of consciousness. Another global state of consciousness is sleep, where the person can be brought back to awareness, but that in principle, during certain stages of sleep, it doesn't have a behavior. It might have an internal life, but we don't know exactly what that is. Another condition would be anesthesia, in which, for instance, we know that the person, without removing the anesthetic drug, cannot be woke up, cannot reconnect. And finally, the study that we more specifically work on is disorders of consciousness. These are people with strong lesions to their brains, either to traumatic brain injury or anoxic insults. And so they typically go into a coma. Coma is a state in which the person is non-responsive to any kind of stimuli, so eyes closed and also non-responsive. There's no kind of stimulation that you can do for the person to react. And then, like, some of these people wake up from a coma and they regain consciousness, but some people wake up from the coma, so they open their eyes and we don't know if they're conscious or not, because like they might have behavior, so they might you know, open their eyes, have a sleep-wake cycles, but there's no way they can respond to our commands. There's no evidence on behavior on them being conscious or not. So we tend to believe that the definition of these global states is supported by the brain dynamics, if we believe that the consciousness is seated on the brain. And this could be described either using, you know, fMRI dynamics or EEG dynamics or any kind of spatial temporal assessment that you can do on the brain activity. At the same time, another group of people, other researchers work on the contents of consciousness. And here the contents of consciousness are going into the question of the specific content of your mind. So whether, for instance, here you're seeing a dog or a rabbit, or you are specifically connected to my talk, or your mind wandering, so you're still awake and aware, but you're connected with a different stream of conscious contents. Or, for instance, if you're dreaming or not while sleeping, so more into the specifics of your contents. And most researchers believe that the same dynamics on brain activity that support one of these lines, a global description of the states of consciousness, should support as well the fine-grained description of what's of the content of your mind. Going into the specifics of these patients and states of consciousness, I believe, and our team believes, that this is both a fundamental but as well a translational project. Why both? Well, first of all, if we're able to disentangle what are the neural mechanisms behind a person being in one state or the other, we will have better descriptions of the neural processes and understanding how the brain is the seat of our subjective experience, but as well drawing a line, creating new tools to draw a line between patients that have subjective experience from those who don't, is crucial because it changes a bit the treatment, first of all, the care and treatment of these patients. So it's not the same thing if a patient is unaware and completely disconnected from the world in terms of pain management and life support than other situations in which you know that the prognosis is better, because the better you are in this simplified gradient, the better the outcome will be. So it's both a fundamental, because we understand what is the basis of subjective experience, but as well a translational project, because there's a clear medical need to create new tools to help diagnose and prognose the state of these patients. So in the lab, we have different lines of research associated to the study of states of consciousness, trying to answer the questions related to what are neural properties and computational properties, what are the limits of conscious processing according to the different states, because we've like this description of global states of consciousness is limited. There should be a dynamical also process in which during the day we are going through different sub conditions, and so what are the limits of processing according to the sub fine-grained description of hierarchical decomposition of global brain states. And then finally, clinical questions regarding whether this patient in particular is conscious or not, whether we can have novel diagnostic tools, and how we can personalize treatment. So we have lines of research working on sleep, anesthesia, in human and non-human primates, and lots in disorders of consciousness. What you see also is that next to each of the publications, we have different neuroimaging techniques associated, and that's also a clear message these days, is that to really understand these conditions, you need to have different methodologies associated, because then you tap into different spatial temporal properties of the brain activity, because each of the neuroimaging techniques, it's focused on the different spatial temporal properties. And also we know from our clinical expertise, because one of the advantages of our hospital, of our center, is that we are the reference for these patients. So we see dozens of patients, so 100 patients or so per year of these. So we know the more of these multimodal assessments that you include, the better your performance is in the prognostication of these patients. So today I'm going to try to give you a flavor of the different things that we do. First of all, most of our work is related to the implementation of neurophysiological signatures of conscious state. This is using EEG to describe the state of consciousness of these patients, to try to prognose if they're going to recover or not. I'm not going to talk a lot about how to perturb the system in the sense of trying to change the properties of the brain to see if that person changed his or her state of consciousness. For that, we use different stimulation techniques. We use a lot of TDCS, TACS, transcranial electrical stimulation, direct current, alternative current. We use deep brain stimulation as well in non-human primates models. I think I'm going to have some of the brain-body interactions work as well, because we believe, of course, the brain is monitoring the external world, but also is monitoring a lot the internal world. So it's monitoring all our internal organs and trying to keep homeostasis on lots of variables. And it's the same organ that monitors the external world and the internal world. So if there are perturbations on the external world, some connection to the external world, that should be reflected as well into the modulation in the internal world. So the idea is that all of the variables from our body should be indirectly formative of a person being conscious or not. So I'm going to deep a little bit onto that, and then I'm going to try to talk a bit more into the fMRI dynamics, and the last 2 lines are basically all together. So trying to see how the dynamics of brain configurations is characteristic of the state of consciousness, and as well how can it relate to the contents of our subjective experience. So I'm just checking how much time I do have. So right, let's go into it. So this is my approach when I started working on this topic, something like I don't know, 15 years, I guess. So there are a couple of theories that are leading theories of consciousness. So basically proposals of what are the neural mechanisms behind our subjective experience. That you probably heard about all the collaboration plus competition between the global neural space theory and integrated information theory that is going on, and all the debate, the trigger, and all the the conflict between the different camps. Though there are lots of oppositions in terms of the predictions, there are as well many common predictions, and those common predictions are related about information. So how information is processed in the brain or any system. First of all, it's clear from all the theories that information should be transferred between brain regions for a person to be conscious or not. That there should be a certain amount of complexity behind this information in order to reflect the external world. So if you need to have a representation of the external world with a certain amount of complexity, that representation should be invariant to that complexity. That you need to maintain that information in for a certain amount of time in order to make operations on that information. And then finally, regarding a more basic type of prediction regarding interaction between cortical and subcortical areas, that certain oscillations should emerge, and those oscillations should be relevant for sustaining a subjective experience. So those were the common predictions, and so the approach was to say, okay, let's try to tackle each one of those and to produce brain markers or bio features, or the way depending on your camp. If you're computational, if you're you come from computational domain, you would say features, and then biology, you say biomarkers. It's the same thing. So, and the idea is not to generate those without an hypothesis. Is that you have an hypothesis of how things should work, and then you generate those computations on your raw data that are reflecting those properties, and then you test whether those are relevant or not. And finally, see if I can try to combine, a bit all of those. How far can I go? So on our first work in that topic, what we did is to compute 92 different biomarkers. Some of them are related to event-related potential. So you have a cognitive protocol behind, and see how the brain reacts to that cognitive protocol. Then you have different frequency bands. Then you have measures of complexity on the brain activity, and then finally some connectivity measures between the electrodes. And what you see on the different columns are the different groups of patients. So on the left, you have the sedative patients, minimally conscious patients. We call it conscious patients, CS, conscious patients. On the third column, now these are called emergent from MCS condition. MCS is minimally conscious state, and then healthy controls. And what we are trying to focus here is to identify which of these biomarkers are showing a phase transition between the first and the second column, because you could have some markers that are more like, gray, they show like of different cognitive properties that go on top of consciousness. If a consciousness is something that emerges at some point, then you should try to find, some phase transition somewhere between one column and the other. And then there are discussions where you should put like the phase transition. Because some people say the minimally conscious patients are probably not conscious. Some of them are, some not, some are not. So maybe within the MCS column, so now the MCS is divided into two, MCS minus, MCS plus. Some would say between BS and MCS. So it's not clear where, if you put behavior on the x-axis and that marker on the whatever marker on the y-axis, where you should be the transition. But we're trying to find something that it's more like a sharp change other than low and steady increase. So another way to see this is like you can compact the spatial properties and then have one biomarker per individual, and then you say how good they are to discriminate. And what we did so back then to characterize that is to compute the area under the curve for each individual biomarker, and then like the different spheres on colors are different contrasts, BS versus MCS, MCS versus CS. So like we should track the red spheres, and the red spheres, the further away they are from the central line, the better the biomarker is. So, there are clear, some biomarkers are really good, spectral measures, information theory, like complexity, and some connectivity measures that are using complexity as well in the low frequency bands are also very informative. So, that was one database of 300 patients. Then we showed that, if you combine all these biomarkers, you get better performance. So great. So the sum of the different biomarkers are better than the individual ones. So they are synergic. But more importantly, the question is whether this is more informative than the behavior itself. So if you just focus on patients that are clinically vegetative, and then you now use these biomarkers to determine whether these patients are, in fact, classified as vegetative or conscious, we see that in 2/3 of the cases, there is a match between the clinical behavioral assessment and the electrophysiological assessment. And there's 1/3 of conditions in which you have a mismatch. And that could be clearly that our system is seeing noise and is not informative. But in fact, if you see the recovery of these patients, when you see something on the EEG that is different, the chances of recovery are double, meaning that in behavioral patients that are clearly undistinguishable from the medical perspective with using behavioral alone, the fact that these biomarkers are pointing in another direction, it's clearly indicative that the prognosis is going to be better. And then we basically replicated this using different methodologies and modalities all over the place for the last 10 years. So it's clear that there is information there. There are two major, like the two consensus papers on the European Union and the US are now pointing that this is crucial to assess these patients. And there is multinational result using these methodologies that is going to be published in a couple of weeks that show that approximately a quarter of what we call vegetative patients are in fact conscious. And then if you use these techniques, you can find traces of subjective experience. That creates a clear, ethical goal for implementing these techniques as soon as possible. Whatever is possible. So in that sense, we have been working the last 10 years trying to simplify the process. Back then we used 256 electrodes. We are showing now that if you can reduce it up to 16, you get a similar performance in discrimination, and that's crucial if you really want to go into the clinics. Then we also show out-of-sample validation. Like, so basically, if you apply the same procedure from data from other centers and from other conditions of recordings, you get similar performance. But that's not enough. So we started a new project recently. It's called Neurometers. And the idea is to make all these tools really available to be implemented anywhere in the world. So basically, the idea is that it's a two-dimensional as a cycle. So what we have is a cloud system in which you can upload your EEG and all the recordings is automatically processed, and then you get all your biomarkers computed semi-automatically. And then there's also, the capacity to have different cognitive stimulations as well provided to the patient using a stimulation box. And then all this information is integrated in the web server, and then you get a prediction of whether this patient in particular it's more similar to one group compared to another, and then you get a prediction of belonging to one condition or the other. And so why this is not yes. So basically, like you can find all the pipelines and everything online, but as well there is a startup like for implementing all these things commercially. This is for some reason. Okay. Anyway, so this is like the report. So far, it's a bit technical. We are working on the display. The idea is that you get an overall vote of all the integration of all the biomarkers, but as well each individual vote, so you can have a bit of a specificity of the condition. And this, it's been implemented in several sites in Europe, in the US already. So that's the EEG. I forgot to say, like, you could introduce me at any point and we can discuss, or we can keep questions by the end. But that's like what we've been doing with using EEG, and we still are doing. But then we try to dig into other techniques to assess these patients. So, for instance, for brain-body interactions, we know that our brain is interacting with different cycles of the body, like the heart, the respiration, the digestive system. They're all providing different types of clocks that are in somehow modulating or affecting our subjective or cognition, subjective experience. Like, for instance, here what you see is if I put you to, if you start looking for Wally, in this game you need to look for Wally. So the moment you start like looking for your target, your heart will start beating slower until you find it, and then it beats faster. So in a way that it's not that the heart is involved in the cognitive process, but the fact that the brain is engaged with a certain cognitive task percolates to other cycles of the other signals of the body, and that could be useful to have evidence of changes in the cognitive processes of the individual. So that would be from the brain to the heart. There's also evidence from the heart to the brain. So according to where the stimulus pops up in the cardiac cycle, the chances of detecting the stimulus changes. So we have a higher, probability of detecting in one part of the cycle compared to another part of the cycle. So the question is, can we translate these ideas to assess DOC patients? So the first task that we did, and this is the work of Fede Raimondo, who was here last year, so I'll probably skip because he might have shown this. So what we did is essentially we deliver stimulus independent of the cardiac cycle, and then what we basically assess is what is the length of this segment, so between a cognitive stimulation and the following cardiac beat, and similar to the previous. So basically the prediction is if the cognitive processing of this auditory stimuli is relevant, then this should be affected, and of course this. So basically different predictions were that, the post interval should be, affected only, that the impact should be, more relevant for global processing of regularities, which are more cognitively demanding, and it should be also more affecting more MCS patients, which more connected to the stimulation than BS patients. So this is exactly what we found. So this is, if you can compare distributions of those post intervals. So this is, sorry, in the right column the post intervals, on the left column the pre-intervals. On pre-interval you have no effect. So like the difference between a deviant and a standard stimulation, I'm not going to go into the details, but basically is the modulation due to the rules on the cardiac cycle. So there's no modulation on the rules, both in BS patients and MCS patients. On the pre-interval, there's nothing on the local deviations, which are very, automatically processed. But on these global deviations, which are really demanding in terms of rule of learning, you see changes on the cardiac cycle only on the MCS patients and not on the vegetative patients. And then what's nice is that if you combine our EEG markers I showed before and these new cardiac markers, you see that the performance of classification is better. Meaning that if you can combine EEG biomarkers, so coming from the brain directly, and activity coming from the body, your performance gets better. So going a bit further into this idea that the more information, multimodal information we can combine, the better we are to classify these patients. So we dig a bit deeper in terms of the naturalistic processes associated to this. So we know that our brains are synchronized when we are connected to the same naturalistic stimuli, to the same stories. So it's classical results from Uri Hasson showing that when you are watching a very engaging movie, the different brain regions are highly synchronized across individuals. When the level of attention or of engagement reduces, so the more boring the narrative, the less the amount of engagement of different brain areas across individuals. That's a clear result on the brain. So the question is, can we do the same thing with the heart? And is it like relevant for patients? So what we did is essentially we put just 2 electrodes on an electrocardiogram, and then you can actually see that in fact there is this heart rate variability, meaning that the amount of the time between 2 successive heartbeats is not necessarily identical. So if you do the inverse of that, that will give you some instantaneous heart rate that you can plot as like nice variations. Then you can spline it, and then you have like a continuous curve of the instantaneous heart rate, and then you can measure the synchrony of this signal across individuals. And that's sort of like the what you get across several individuals. In fact, if you average the amount of synchrony that one individual has to the rest of the group, it could be 0, above 0, etc. So the first result is that in fact, when you are listening to a story, the level of synchronization of this heart cycle across individuals is highly significant. So in fact, if you shuffle you have several snippets here of the story. If you shuffle the snippets across individuals, the synchronization completely disappears. There is a clear increase of the synchronization when you have the original versus the permuted segments. If you average the different segments, you have significant synchronization across individuals in more than half of the individuals. That will be the first result. So it's clearly that the narratives are synchronizing the heart activity. It's not that our hearts are beating at the same time. It's not that, like the fact that we are watching the same movie, our hearts will beat on the same time. It's that we will increase our heart activity or heart rate and decrease at the same time, independently of the baseline heart rate of each one of us. We modulated the attention of those streams, of those narratives, by asking the individuals to do counting backwards on steps of 7, starting at 1,000. So it's very demanding. So if you ask someone to do that and you put him to watch a movie at the same time, there will be a conflict of attention. So if you do that systematically, the level for the same stories, these are 5 different stories, each sphere is an individual. So systematically, the level of synchronization that the individuals have is decreasing if you perturb the attention. If you average the 5 stories, all of the individuals except one are decreasing the level of synchrony to the rest of the group. And then also the level of synchronization of the cardiac cycle predicts if you're going to remember the story or not. So basically, we posed questions after the stories, more complicated stories, and the amount of synchrony. And blue these are the distracted individuals. In red, the attentive. Clearly, there's a difference there. But as well, even within, if you consider both groups, but also within each of the 2 groups, the memory performance is clearly related to the intersubject synchrony. So what about patients? So this was a proof of concept. So you have many patients, but the interesting thing is that only 2 patients show a significant intersubject correlation. So basically, the correlation of the patients to the healthy group. So we use a healthy group, the activity of the healthy group as a measure of engagement with the story. So only 2 have that significant, and those were the 2 that recovered. And in fact, if you correlate the level of synchrony of these individuals to the indemnity of the white matter tracks, you find that both are really related. So it's really, I think, interesting tool because compared to complex EEG assessment, fMRI, PET, or the different types of techniques that we use on these patients, so this is something, an entry point for assessment or additional evidence for these patients that could be easily implemented anywhere in the world. That's where we're going. Now we are in this line of work. We are trying to disentangle the spectrograms, like some parts of the spectrogram. Is it the predictability in terms of the language? So there are many layers you can decompose a narrative. So the idea is to see what are the different domains of the different features of the different of the narrative that are behind the intersubject correlation.
Quick question. You see that the healthy controls have quite some variability. Yep. I wonder if then on group level it's good effect, but does this translate well on individual level?
So first of all, here you see that at the individual level you have, for the patients you have a measure, and the 2 that were significant were the ones that record. This is very, very slow amount of prediction. Of course, you see a lot of variation, of course, but then if you use the same individual as a reference, all of them are decreasing when you disconnect when you contrast the distracted versus attentive. So there seem to be modulations of this. First of all, this is very indirect measure because we are measuring the modulation for the cardiac cycle. So if you associate this now to the EEG as well, then you have a measure of synchrony on the EEG, it gets better. So I believe that the advantage here with the different measures is that you will tackle different, hierarchical properties of the engagement. So with the EEG, you have like fast engagement, some, things associated with fast engagement. With cardiac cycle, with the slower processes, and perhaps if you go in deeper, like using respiration or others, you can go into more slower, even slower processes. There were other questions in this.
Have you matched the results with the personality traits of the patients?
No, but the patients is a bit difficult. So
cognitive engagement, yeah, it depends on their background, depending on the films.
No, so we didn't do that. But there was a paper like last week that I found it like super interesting. So there's people working, a lot of people working on social interactions and the way we make decisions and the way we. So what they did is basically they show a very ambiguous movie to people, and then you have like zero synchrony across individuals. And then what they did is they separate into groups the people, and they then put it to discuss about what they have seen. And then you have reports of how good they were reaching as of consensus or not within the groups. And the groups that reach certain consensus when they re-saw the movie or re-saw other movies that are with a similar type of ambiguous processes, they have very high synchrony. So it's really, I think, taps into the way we interpret the world. So
maybe more the emotional engagement than the cognitive one. But it is
how you associate the two?
Just the cardiac rate. I mean,
probably. That's why we want to also decompose a story into different properties of it, or the narrative, and see what's really behind. What we found so far with EEG is that, in fact, all the dimensions. So if you remove systematically. So one thing you can do on EEG, but also here, is to compute transfer functions. So you have like some property on your history, and you compute transfer functions of that property into your time series, and you can systematically remove one by one. So you see, okay, I'm going to remove the low-level properties of the story from the EEG and see if there's a synchronization still. So systematically, if you remove one by one, you still see a synchrony. So it's probably an interaction of all the dimensions, dimensions of the narrative that are really behind the way we interpret. Emotions clearly play a role, and it's probably on a much higher level of interpretation, but even lower levels are playing a role that are probably associated to emotions. So it's an interesting line of research, I think, and opening again, avenues of developing new tools to assess these patients, but as well understanding a bit of the neural processes behind these interesting cognitive properties. So let's go into the last line that I wanted to mention. So regarding the fMRI dynamics, and here, the prediction is a bit more basic in the sense that we try to go into the William James description of stream of consciousness, of our sequence of the way we interpret, our subjective experience as a continuous stream of subjective processes. And so in a way, like this, our conscious processing is reactive or the dynamic nature of the environment. So in a way, the more complex the environment, the more complex is the dynamics of spontaneous activity. And so we try to tap into that prediction using a model of non-human primates and anesthesia. This is a collaboration, a long-run collaboration with visual AI and neurospin. And so what we did is basically what we have is this is the dynamics of brain activity across areas, and you see how one area interacts with another. And then typically what you do on resting state analysis is to average the activity during the whole session, so you don't care about the fluctuations. But in fact, there's a lot of richness here. So there's one area might interact with another, but then stops doing it, and then interacts with another, and then reconnects with the original area. So there is a lot of richness there that is disregarded. And so we wanted to include that into the analysis. So what we did is to to try to decompose the signal into a series of modes that are more frequent during the session. So we have sessions of monkeys that are awake or anesthetized, and so what we have is a sequence of modes, and then what we do is we try to do unsupervised learning of the major brain states that are occurring across all the sessions. And then what we see here is, the different, like in this first study, what we see is that there are, certain modes are highly frequent, highly repetitive. And in order to interpret them, what we did is to order them according to the similarity of those maps to the structural connectivity. So on the left, you have things that are less similar to the structural connectivity. On the right, things that are more similar to the structural connectivity. And what you see is typically said on the left, you have anti-correlations, long-range interactions. And on the right, what you have is only a few connections, a very sparse matrix with only positive connections, and the overall intensity of the interactions is very small. So we have sort of like an exploration space of states of configurations, and we try to characterize each session by the amount of the frequency of occurrence and the transitional probabilities of these different states in each session. And each session is in a monkey either anesthetized or awake, so it could be characteristic of the global state of consciousness. What we saw is that, in fact, when the monkey was awake, the exploration is more or less flat. So the monkey is exploring all the different configurations in a very similar frequency. So on the x-axis, you have the similarity score of the functional maps and the structural map. So on the left, you have the anti-correlations, and on the right, you have the sparsity, sparse maps. And so you have a really flat interaction here. And then once the monkey is either moderately sedated, moderate, even like extreme non-responsive, or deeply sedated, you have a clear disappearance of all the states on the left and a predominance on the states on the right. So basically, in one line, what happens when you exercise is that only the wires remain. So only the interactions that are directly driven by the wires remain. So that's what we found back then on monkeys. And then the question is this the case as well with humans? And we did a multicentric study using the same analysis, fMRI dynamics, on DOC patients, disorders of consciousness patients, on 4 clinical sites. Lots of recordings. These patients are really difficult to record using fMRI. That's why you need to do multicentric studies as well. Gives you power in terms of the multi-site validity of our results. So what we got is, this time we describe it on 4 patterns, but you get the same thing, you do it on 7 or in 3. So basically, again, the anti-correlations, long-range interactions on the left, sparse interactions on the right, the same way we order the patterns. And then what we saw is that the frequency of this pattern here, which is the pattern of anti-correlations and long-range interactions, was very low in vegetative patients, increases on MCS, and systematically increases again on healthy controls. The distributions in 3 sites, Paris, Liège, and New York, is very similar. So same in all cases. The 2 in the center, we found no differences between the 3 clinical groups, but the one with the sparsity, in fact, you have the reverse pattern. You have very frequent that pattern on vegetative patients, and then decreases systematically as you go up on the clinical states. So if I plot this using the same procedure we use on monkeys, we get a more or less flat type of relationship for healthy controls. deeper slope for MCS and an even deeper slope for vegetative patients. Just to remind you, this is what you get on monkeys. So 2 species, 2 ways you can lose consciousness, the same fingerprint. We are excited about this because we think it could be something a bit of universal and nice, interesting to explore. And in fact, this is very recent. There are people that are proposing using deep brain stimulation to treat these patients. And so we did a study on monkeys here again, and the monkeys anesthetized. The monkey has an electrode in the central thalamus. What you see is here. This is the awake monkey. So the flat type of relationship is if you turn on the stimulator. So you get a retrieval of the relationship of the occurrence of different brain patterns when you stimulate. If you stimulate the electrode next to it, nothing. If you stimulate exactly the electrode on the central thalamus, you get this change of the exploration of brain dynamics. The interesting thing is the monkey still behaviorally, it changes a little bit, but it's not very dramatic. But it does change the dynamics of brain patterns. And that would be a bit suggestive that, in fact, you're going into the right direction. And you have more supporting evidence from the cognitive processing of stimulation during stimulation and the EEG as well. And now we are trying to move this forward into non-invasive type of stimulation using ultrasound-focused stimulation. So you can focus ultrasound stimulation to anywhere where you want on the subcortical structures, and see if we can use that as well to change a bit to see how the dynamic changes. That's also very promising in a sense in the direction of therapeutic interventions. And before I finish, so I also, mentioned the idea that we want to measure or assess whether these brain dynamics these processes of brain dynamics is relevant for our, not just the global state of consciousness, but the particularities of our subjective experience. And what we basically show, this is the work of Başak Turker that she did the PhD with us. She started during the pandemics at the beginning of our PhD. We have very ambitious goals. We had to work with previously acquired data because all the acquisitions were not possible back then. So what we did is to do the analysis of brain patterns during movie watching. This is data from Adrian Owen and Lorena Nasi. Adrian is in Canada, Lorena is in Ireland. So what we have is people like watching a movie, regular movie, or you scramble the movie, or you do resting state, and what you observe is that the patterns, now you have like a hierarchical decomposition of these complex patterns of the ones with anticorrelations into 2 different subtypes of patterns with anticorrelations. One specific for the movie condition, the other specific for the resting state and scramble condition. So in a way, you have the same frequency of kinds of patterns, but it's one that emerges more frequently when the subjects are engaged with a movie, and other is more frequent for the other condition. And then most interesting thing is that the level of similarity of the sequence of the patterns itself, it's very specific it basically synchronizes for movie compared to scramble condition or resting state. So basically, you might have the same proportion of patterns, but they align when we are engaged with the same dynamics of narrative, meaning that the occurrence of these, so it's not just a frequency, but the specific occurrence of when it's somehow like associated to our subjective experience. And then the more ambitious experiment came later, and what we did is basically do a protocol of subjective of perceptual detection. So you have tones that are really masked by some background noise, or the scanner noise as well, and basically the subject whenever he or she detects the stimulus, it presses a button. So you can play a bit the relationship between the tone volume and the background noise, such that you have a perceptual curve. It's more or less centered at the chance level, and the prediction we wanted to test is like if you have a perceptual pattern, a perceptual curve like this, when you at the center of the SNR level, you have 50% chances of detecting. So the question here was, according to the ongoing brain dynamics, would we see increase of the detection rate or decrease? So basically a shift of the curve to the left or to the right, according to the ongoing brain dynamics before the stimulation. So this is what we got overall on the detection rates. So like a really nice perceptual curve with a 50% chances with a central signal to noise ratio. And we have, according to the ongoing brain pattern, we have a shift of that curve for the patterns that have anti-correlation. So the patterns on the left. So basically, if we focus there, what we see is basically on the pattern one, the one with anti-correlations that is more associated to subjective experience, the chances of detecting a stimuli, so your engagement to the subtleties of the external world, it's higher. So it's a very small effect. It's a small effect, but it's clearly there. So it's really, our brain dynamics, the specifics of our brain dynamics is really pushing towards detecting or not, or the engagement that we have or not to the external world. So just to wrap up, so we show generalization and out of sample validation of EEG markers of consciousness, and that's been translated to automatic web-based pipeline. New exciting biomarkers, brain body biomarkers. These are relevant for seeing how the interaction between our brain and the body reflects our subjective experience. The clinical validation of dynamic of global patterns, of fMRI global patterns as a marker of consciousness, and how interventions of that potentially, one line of research we want to push is that now you can have techniques of neuromodulation, providing stimulation, reward, etc., according to the ongoing brain dynamics. See if we can use that as well as a way to change the state of consciousness. And finally, the idea of using new therapeutic strategies using either non-invasive or invasive stimulation techniques on these patients. So with that, I thank all the people that I work with, including Lou there on the back, where we are, part of a common international program for trying to extract biomarkers from modeling of this brain data. We'll see if Lou presents a bit of that. And we have a couple of open positions. If you're interested, don't hesitate to come chat with me or write me afterwards. Thank you.
Questions? Questions about the first part. Have you tried to match EEG and functional imaging with brainstem evoked potentials, like auditory evoked potentials, which they don't need the patient interaction and they are more robust when anesthetized? It's easier to
absolutely. So in fact, all our database, it's in fact not resting state. It is a auditory protocol, because we use what we call the local lower protocol. It's a protocol that has a series of auditory tones, and you have early potentials associated to each of the tones. Then you have late potentials associated to the rules either local or global rules associated to those tones. So you have, we analyze P100, mismatch negativity, and P300 associated to these sequences. And this is very, very, very informative and complementary. The faster potentials that are,
The solar 300, but the auditory will be even easier in setting.
Yeah, absolutely. It's easier and not. So what we find is that when you have a positive result, so basically a mismatch negativity or a P300, this is highly informative for the state and evolution of the patient. If you don't find it, it doesn't say anything because there's so many things that can go wrong. So in terms for the positive predictive value, ERPs are very good. In terms of negative predictive values, the other metrics are better.
To confirm, as example of a brain death, when the transplant team is waiting and you have to confirm.
Absolutely, it's part of the Maastricht rules these days.
Potentials too, not only EEG, because it's much more sensitive to anesthetics and many other confounders.
Yep, so we use that a lot, and we have a couple of papers showing the predictive, like I said, the positive predictive value, it's extremely high. If you find a P300 on these patients, the chances of recovery are almost 100%.
Last part, the fMRI states. What were the temporal dynamics there? Like, how long did they stay in each state?
It's one of the properties that is relevant for assessment. So what you have, the higher the state of consciousness of the patients, is a more metastable condition. So they might stay longer within one global pattern. Typical distribution of those values is the order of 10 seconds, 12 seconds, etc. When you are in a vegetative state condition, they only stay for a couple of TRs, 4 to 5 seconds. So the depth of this metastability seems to be a good factor. Also, we are now trying to interpret this beyond because of course the reduction into global patterns, it's limiting. It's nice because it gives you a framework to analyze, but it's limiting in terms of the real ongoing dynamics. So we are trying to see this in a much larger space as a random exploration, a random walk on a space with potential level flights. So that what they typically do, a healthy individual, is to stay to explore an area with more stable condition, but also make like huge jumps that you don't see on vegetative patients or minimally. So there's a lot of richness on the dynamics that I think it needs to be explored.
In this early part of your talk, you had this big list of these features, the spectral and then information theory. Just can you walk me through a little bit, like what these features are?
So basically, what we did back then, which you have to keep in mind that it has evolved a lot. That field. So with this back then, what we call is feature engineering. What we call it. It's basically you have a predefined set of ideas and you transform those ideas into features, and then you see how they good and bad they perform. Nowadays, it's all bottom up in terms of the data. You don't care. You can have like these nice pipelines. You compute 2,000 features and okay. Anyway. So what we did back then was to propose a taxonomy of biomarkers. That taxonomy basically split into resting state biomarkers and evoked biomarkers. That's your first split. So resting state basically provides a response when you average several epochs. The drill. But basically, back then it's basically mismatch negativity, P300, P100, the variability of all of those in terms, also in terms of time and amplitude and spatial distribution. So that's one domain. The other domain, in terms of resting state, we split into spectral decomposition using different compositions, so power and different frequency bands, either normalized or not to the whole frequency power. Spectral summaries, so basically seeing where the spectral distribution splits in half in terms of power, MSF, SF90, SF95, and the complexity of the spectrum using spectral entropy. So those the spectral ones. You can add more. New features are proposed systematically. What we basically want to say now, 15 years later, is that what we basically proposed back then is a way of thinking, and then you can now change your recipe a bit, like add more or remove some of features because they are no longer relevant, and then like the pipeline still is valid. So again, spectral, and then the complexity measures are permutation and complexity, and Kolmogorov complexity, basically permutation entropy. It's sort of like a transformation of the signal into a series of symbols, and then you measure the complexity of that sequence of symbols. And then Kolmogorov is basically the idea of the algorithmic complexity. So map how much information you have in that signal by compressing it. And then finally, we did something like 20 or 30 different connectivity measures. The one that worked best back then was one that we proposed as well, which called weighted symbolic metric information, which is basically, again, symbolic decomposition of the signal and then doing a bivariate analysis of the sequence of symbols instead of working with the raw signal. So there's advantage there of two advantages. One is this time series are very noisy because the recordings are extremely noisy on the bedside. So the symbolic decomposition, enhances the SNR by losing information, but enhances the SNR. And the second is that you tap into nonlinear interactions, which seems to be more relevant in this case compared to other conditions. Now, people who are then use the same metrics to analyze what you see on comparing PLI and WSMI, and then you're basically tapping into different properties that might be relevant in one condition compared to another. But then you can add new stuff and then rerun and see what it's your new recipe. There.
A question about the DBS.
Yep.
So when you stimulate the thalamus, that you see that there is an increase in the number of states that are visited.
Basically, the number of states is always the same because this is a space where we use to describe the exploration. So with different kinds of analysis, this analysis wouldn't reflect the fact that there are more states present. It's just with the space of states that we use to describe, you have a more complex exploration when the monkey is awake. That complexity of exploration decreases when anesthetized. And here, in a way, mirroring the recovery of consciousness when you remove the anesthetic, when the monkey is still anesthetized, so the drug is still there, but you put the stimulator, you recover the complexity.
Because I was wondering that maybe there is an increase like in entropy, but it's not organized like it would be in a awake state. It might have no content of like information, but if it's the same states, it's
no more. Then again, this is just a frequency. Frequency, like your occurrence, is one thing. The order of how you change the things, and how relevant that would be to have a subjective experience is still open to debate. It's clearly that he probably wasn't as connected or conscious as in a normal condition, because it doesn't behave the same way. But the evidence on the overall frequency of occurrence points towards what you observe when the monkey regains consciousness. Then again, it's a question of the hard question of consciousness, so we really don't know.
If there are no